Hello, my name is Carol Tamminga. I'm a psychiatrist, a clinical researcher at the University of Texas Southwestern Medical School in Dallas, Texas. I've been invited to prepare a plenary plenary talk for this lecture, and I'm terribly honored to be with you and to share some of my thoughts with you about psychosis and schizophrenia. With that, I would like to share my slides with you and start the presentation. The content of what I'm going to talk about today will be a talk about a BSNP consortium. I'll tell you what BSNP means in a minute, but we have a focus on developing biomarker-based treatments for psychosis from a BSNP data set, which I will also show you. The question with these data is, are we ready yet? Are we ready yet? Psychosis in schizophrenia is something that all of you know very well. You have many patients who have this illness. You know, the engraving on the left is an engraving from Bethlehem Hospital, and it's one of my favorite engravings. The word Bedlam came into the English language because if you say Bethlehem very fast, it sounds like Bedlam, and it describes that behavior. You can see what psychosis looks like. Then in 1996, one of the magazines in America, not a scientific magazine, the National Geographic, wrote an article on diseases of the brain. It was the first time that schizophrenia was called a disease of the brain in the American public literature. You can see, although there aren't nearly as many people around, the setting is quite familiar. And it's obvious that for centuries, we've really failed to understand and cure schizophrenia. The Bipolar Schizophrenia Network for Intermediate Phenotypes, what I'm going to call BSNP throughout this study, is a group of investigators, and you can see us all here, across the United States. And our intent was to dimensionally recruit a psychosis sample, a sample of people with schizophrenia, with schizoaffective disorder, and with psychotic bipolar disorder. And we recruited two different waves of these, one in BSNP1, the first of the studies. The N's are fairly high, giving us plenty of people to work with for computational analysis. BSNP2 was the replication study, and I'll show you some data from that study as well. These are my collaborators on all of the BSNP work. Dr. Clements, Dr. Gershon, Dr. Keshavan, and Godfrey Perlson. And this comprised the BSNP group in the United States. Now, I want to share with you what our rationale was at the beginning of the study. So we knew there was a disease called schizophrenia, another one called schizoaffective disorder, another one called bipolar disorder. We recognized that there were gray areas in the middle of these, but what we really wanted to do was to look at intermediate phenotypes. You can call them biomarkers, you can call them intermediate phenotypes. We wanted to measure many of these intermediate phenotypes, find intermediate phenotypes that fell on each of the diagnoses, and then find the genes that were attached to the intermediate phenotypes. It was thought in those days that genes would be more easily found connected to intermediate phenotypes than to diagnoses. Now, this was where we started. The plan was rational. It was an interesting way to start a study, but I just wanted to tell you that despite it being very rational, in fact, it was all wrong. But let me tell you what we did. So first, what we did in the BSNP study really depends on these two factors. We had large numbers of patients, we had large number of patient volunteers, and we had a comprehensive set of biomarkers, and I'll show you that in just a minute. In all of the groups, whether we divided the sample by a conventional diagnosis or whether we divided the sample by biotypes, which was biomarker-based units, we had about 500 individuals per group. For each one of those individuals, we had a complete clinical characteristics. We did all the demographics. We did a SCID diagnosis. We had different rating scales, the PANS, the YMRS, the MADRS. We did the Birchwood Social Function Scale and some other smaller scales. And then for biomarkers, we collected biomarkers in cognition, neurophysiology, imaging, eye tracking, blood-based biomarkers, and then we have stem cells in addition. For cognition, our basic measure was the BACS, but in addition to the BACS, we did some we collected some additional scales. For neurophysiology, we had both resting state data and we had evoked potential data. We had auditory data, we had visual data. For imaging, we collected structural imaging and we collected resting state fMRI and we collected DTI. For eye tracking, we had both saccadic and anti-saccadic movements. And blood, we collected blood for different immune biomarkers and for DNA. And we're still now in the process of working with these stem cells. What we wanted was to get a comprehensive set of biomarkers that was not just determined on what we did in our own laboratory, but that was really representative of the kinds of biomarkers that had been discovered and reported in the past to be abnormal in psychosis. You can tell we had a lot of data to collect. We collected them in two sessions, well, the first 5 years for BSNP1, the second 5 years for BSNP2. Let me tell you what we found, really, in people using this cartoon. We have a lot of data in addition, but using this cartoon, we had people with schizophrenia who were very heterogeneous, schizoaffective disorder, psychotic bipolar disorder. We found no differences of all of those biomarkers I just mentioned. We found no single biomarker that fell strongly enough on each one of these diagnoses to use them as a diagnostic biomarker. There are some biomarkers that fell with enough to be significant, but they were not strong enough to be diagnostic biomarkers. So what we did instead was to just combine all of these conventional diagnoses into one group of psychosis. All of these people, we had a comprehensive set of biomarkers. So we used those biomarkers. Chiefly, we used sensory motor reactivity and we used cognition. And we used those two sets of biomarkers to divide people up. And we found some fairly homogeneous, more homogeneous groups than the psychosis group. We named these biotype 1, biotype 2, and biotype 3. Biotype 1 and 2 both had low cognition, but biotype 1, in addition to low cognition, had very low EEG power. They had low resting state, they had low evoked potential. Biotype 2, instead of having low EEG, had high EEG, both resting state EEG and evoked potential. Biotype 3 had fairly normal resting state and evoked potential. Most of the cortical, neocortical reductions from healthy controls, the biggest reductions were in biotype 1, and they were pervasive throughout the whole neocortex. In biotype 2, we saw also reductions. They were a little, they were lower reductions, less reductions, and they were localized to specific regions, mostly localized to the frontotemporal regions. In biotype 3, we found really almost no cortical loss, just in the core limbic structures. We found most of the highest number of relatives with schizotypal disorders was in biotype 1, and there were some in biotype 2. So here's really a list of what those characteristics are that describe biotype 1, biotype 2, and biotype 3. In order to get to these biotypes, we took all of the biomarkers. There were probably 50 or 60 biomarkers. We condensed them to 9 different biofactors. These were distinctive dimensions of biomarkers. And then we used those 9 biofactors. And I'll show you those factors in just a minute. Those 9 biofactors to do our calculations. One of the, so I told you some of the characteristics of biotype 1 and biotype 2. Biotype 3 had really the least positive biomarker characteristics in the whole group. And we couldn't really figure out what might be the issue until one of the people said, well, maybe these are the potheads, as we call them in the United States, which are the high cannabis users. And when we took a look, in fact, people in biotype 3 were the people who had the highest overall lifetime cannabis use, but they also had early cannabis use. Now, this was a quite fanciful approach to really looking at the intent that we had was finding out biological characteristics of people with psychosis, as opposed to, in contrast to the phenomenological characteristics that we looked at with a conventional diagnosis. So we certainly had to replicate those findings, and I'll show you in just a minute the data from BSNP2, but the replications were really, so they even surprised us, they were so high. Then we had to show that this biotype classification was good for something. What would be the most immediate question is, what are the treatment implications of these biotypes? And that's what I'm going to talk about today, the treatment implications. We are doing genetic characterization. We're not done with that, so I won't talk about that today. I wanted to show you that in the conventional diagnoses distributed within the biotypes, this is biotype 1, this is biotype 2, this column is biotype 3. What the dots in orange are the dots with schizophrenia, fairly equal over biotype 1, 2, and 3. The dots in this in turquoise and green and turquoise are schizoaffective disorder, and the dots in purple are psychotic bipolar disorder. Psychotic bipolar disorders have a significantly higher cluster in biotype 3, but they have a distribution across all of these. This is a replication data that I would like to show you. This is BSNP1 from the early part of BSNP2. We still have additional cases to run in BSNP2. We're not quite done with it yet, but this is a BSNP1 versus a replication sample. Here you see on the left, you see the volunteers stratified by conventional diagnoses, and the biotype, BSNP1 is the solid line. BSNP1 shows you the differences across the 9 biofactors, the cognitive biofactors over here, the EEG biofactors over here. It shows you the standardized units in each of the diagnoses. What you see, first of all, is that the normals are up here, and the schizophrenia, schizoaffective, psychotic bipolar do not distinguish themselves very well across any of these markers. But you can see, if you compare the solid lines to the dotted lines, that the replication is very high, even in conventional diagnosis. If you look at the figure on the right, you'll see BSNP1 versus BSNP2, and you'll see a distribution by biotypes. We reclustered them to B1 in red, B2 in blue, and B3 in green. Again, the normals are in purple. You can see that the B3 is awfully similar to normals, both in the cognition, a little bit different, both in the cognition area, and really with some decrease in some of the electrophysiologic measures. But B3 is still close to normal. B1 and B2 have both low cognition. They have low scores on all of our cognition measures, but they replicate from B1 to B2. In the electrophysiologic measures, B1 is very low, and that replicates between BSNP1 and BSNP2. B2s, biotype 2s, are very high, and again, that replicates between BSNP1 and BSNP2. And you can see that these replication figures, the interclass correlations, are between 0.95, 0.98, sometimes 0.99. So now, if we're thinking about basing treatments on biomarkers, biofactors, or biotypes, we have to figure out how to do it, and we have to test the hypothesis about whether this would bring advantage to our treatments. We've been focused on finding biologic features of psychosis for so many years that we've maybe assumed that this would bring it an advantage, but it's clearly a hypothesis. And I'll tell you a little bit about our plans to test. I'd love to tell you about that. What I'll talk about today is really 3 different studies that represent 3 different approaches that BSNP will start with. In the first study, we have the hypothesis that biotype 1, this is the low EEG power, predicts responsiveness to clozapine. And we'll do this study over the whole BSNP consortium, and the study will be funded by NIMH. So what do we predict in this study? I first want to show you that our biomarker outcome is low intrinsic EEG activity. You see low intrinsic EEG activity here. Biotype 1, you see here what this intrinsic EEG activity looks like in healthy people. You see then what the intrinsic EEG activity looks like in B1 versus B2. So this particular study will take biotype 1 and biotype 2 and compare them with clozapine. So in biotype 1, we'll treat a group of people with clozapine versus risperidone, and we'll do the same thing in biotype 2. We want to show that biotype 1 responds to clozapine and not to risperidone, and biotype 2 responds neither to risperidone nor to clozapine. Now, this is not a standard antipsychotic response. This is a response of the extra added value that you get with clozapine treatment. We know that both clozapine and risperidone have antidopaminergic characteristics, so we will see that, but we'll see an added response. The hypothesis is that we'll see an added response in B1 versus clozapine versus risperidone, and we'll see no response in B2. Now, how did we come to this idea, really? We looked at all of the drugs that were used, the treatment compounds that were used in the BSNP1 people, the 2,000 people. And we saw that clozapine was unique amongst the antipsychotics in increasing intrinsic activity, intrinsic EEG activity, and therefore it normalized intrinsic EEG activity in B1. Now, if you'll look at this, the top graph a minute, a biotype 1 is drawn in red and pink, and biotype 2 is drawn in the blues. So this first bar is people with biotype 1 people who are not on clozapine. And you see they have a decrease in EEG power, in intrinsic activity. Once we looked at the biotype 1 people who were treated with clozapine, we saw not only an increase, but we saw a normalization of that. In biotype 2, you see the same increase, but because biotype 2 already has high EEG activity, clozapine it generates a quite high EEG activity in the biotype 2. This happens in both the theta range and the alpha low beta range. Risperidone, which we're using here and we will use in this study as the comparator, both on and off risperidone, the biotype 1 demonstrates low and equal intrinsic activity, and biotype 2 demonstrates a high activity in both conditions. So we have a model that we will use to explain this finding. This is the attractor network model in clozapine. In normal, here's drawn just as a way to model it, a healthy attractor network. An attractor network is a group of neurons that fires together around a single task. It could be a memory, it could be a motor command, it could be thinking, but these are neurons that are trained to act together. What the EEG characteristics of biotype 1 suggest a very compromised attractor network, which is very low overall. This is biotype 1 on any antipsychotic drug. This is our model of what biotype 1 will look on clozapine, whereas instead of having just very low intrinsic activity, it will have all of its attractor network normalized. And one of the reasons we really think that clozapine will do this is because of the broad, the complex neuropharmacologic actions that clozapine has. It's a ligand at the dopamine receptor, serotonin receptor, muscarinic, NMDA, GABA receptors. It has very, very broad action. And one of the things that's really that's really known about these attractor networks is that they're kept in balance by by a complex group of neurotransmitters, not just by only one. So that's our model, and this is how we'll test it. We'll recruit about 320 people. These will be only biotype 1 and biotype 2 cases. We'll clean up their medications, and then we'll take biotype 1 and biotype 2 and cross them over either to clozapine or to risperidone. So we'll have 4 different groups: biotype 1, clozapine, risperidone; biotype 2, clozapine, risperidone. And then lastly, we'll contrast clozapine and risperidone. We'll do safety monitoring, of course. We'll do clinical outcome assessment, and we'll do biomarker outcome assessment. We will hypothesize that a biotype 1 treated with clozapine will do the best, and that the added value, the added additional antipsychotic action in biotype 1 will correlate with the increase in intrinsic activity. So this is one fairly, is one kind of study that we will generate from the BSNP group. Now, there's another study that we're going to do, which is a new drug study of this new drug, KMD-001, focused on cognitive dysfunction. And this drug is a CAT2 inhibitor. This study is being done by the BSNP consortium, along with Robert Schwartz. Dr. Schwartz has really looked at the tryptophan pathway for his whole career and identified abnormalities in psychosis and schizophrenia. And his abnormalities and his drug candidate, which is the KMD-001, really captured the interest of a Swedish drug company, Kinexus Medicine Development. So with these 3 of us all together, we will approach this study in schizophrenia. So one of the things that's difficult in schizophrenia, if you have a new mechanism, is selecting a sensitive population. Of course, if this can is all of what we call schizophrenia, we know that almost all of schizophrenia is sensitive to dopamine receptor blockade. What we have in this particular study is we have a very small sample within the sample of what people call schizophrenia. We have a very small sample that we postulate ahead of time will be sensitive to kynurenolic acid. So how do we find this small sample so that we can direct our novel drug to this small sample and not direct the novel drug to all of the schizophrenias where we know that the effect will be diluted? Let me start with Dr. Schwartz's characterization of the kynurenine pathway in schizophrenia. Here's the pathway in normal people, the pathway in schizophrenia. In normal people, this is really oftentimes called the tryptophan pathway because it starts with tryptophan. Tryptophan is metabolized really to L-kynurenine, and L-kynurenine, the pathway diverges. There's one pathway that goes that goes on into the astrocyte. There's another pathway that goes on into the microglia. Of the pathway that goes on into the astrocyte, the enzyme that carries the L-kynurenine on is KAT. There's 2 different enzymes, 1 and 2, and kynurenic acid is the outcome factor. Kynurenic acid is known to be a negative modulator of the alpha-7 acetylcholine receptor and a negative modulator of the NMDA receptor. In schizophrenia, there is a lesion in schizophrenia, and I'll show that to you in just a minute. The enzyme KMO that takes L-kynurenine down into the microglia pathway is deficient in schizophrenia. The deficiency of this enzyme pushes more kynurenine into the kynurenic acid pathway and increases the concentration of kynurenic acid, and that would increase the negative modulation of the alpha-7 cholinergic receptor and also of the NMDA receptor. And it's Dr. Schwartz's hypothesis that this action on these receptors is deleterious to cognition. This kind of pathway and this kind of a This kind of the basis for this research has been done for several decades by Dr. Schwartz, both in animals and animal models and in human tissue. Let me show you a little bit of the human tissue data. This is schizophrenia postmortem tissue, and it's prefrontal cortex. And here, Dr. Schwartz measured KMO. This is the enzyme activity that I showed you was deficient in schizophrenia. And this is kynurenic acid. This is the product of L-kynurenine. And as I showed you before in the diagram, in schizophrenia, both in BA9 and in BA10, there's a decrease in KMO activity. There's a decrease in the KMO protein, and there's a decrease in the activity, which Dr. Schwartz has showed in other studies. And that increase correlates with an increase in kynurenic acid, again in the same areas, in the same samples. The gene in human postmortem tissue, there's a KMO SNP, and it's only the CC genotype within that SNP that's associated with reduced KMO mRNA. In control samples, in control human postmortem tissue, whether you look at the TT plus CT genotype, or whether you look at what we now would call the deleterious genotype, the CC genotype, the KMO mRNA expression is equal. In schizophrenia, the TT-CT genotype expression is equal, but the CC genotype is reduced in people with schizophrenia. And that's important because we use this fact to get to our biomarker fingerprint for high kynurenic acid. What we wanted to do was really to take a group of people with psychosis or schizophrenia. We wanted to test them for high kynurenic acid in brain tissue, and then we wanted to treat them with a drug that would treat kynurenic acid. We will, in order to define this biomarker fingerprint, we started with biotypes 1 and 2. Those are the low cognition biotypes. Out of these biotypes 1 and 2, we selected individuals with this vulnerable deleterious KMO genotype. We took all of these individuals and we used them to define, we used the BSNP database to define a biomarker fingerprint that correlated with this genotype, and I'll show you that fingerprint in a minute. And we wanted to do this as a way of identifying high kynurenic acid individuals. Why don't we just use this single genotype? And the reason is because we think that there's probably several different ways to get to elevated kynurenic acid in addition to this single genotype. So we wanted a kynurenic acid a biomarker fingerprint, so we could determine who really had high kynurenic acid. So we did that. We found a biomarker fingerprint that was associated with cognition, electrophysiology, and anatomy. And let me show you the data in the next slide. We found this combined fingerprint made up of cognition neurophysiology biomarkers, cortical thickness biomarkers, and cortical surface biomarkers. These cortical surface, all of these biomarkers are shown here with their correlations. And if we use all of these together, we can find a rather high probability of finding a group of people with psychosis and poor cognition, and at the same time high kynurenic acid. This is the CT-TT genotype, and this is the CC deleterious genotype. So this is the genotype of people with high kynurenic acid. If we pick the upper distribution of people with this deleterious genotype, we'll have only a little contamination with a non-deleterious genotype. And we think that if we use the drug, the anti-kynurenic acid production drug, if that drug is active, we'll get a chance to see it. So how do we find this high kynurenolic acid, or this drug responsive group? We take all B1 and B2 psychosis individuals, so we have low cognition people. We identify those individuals with this biomarker fingerprint that I just showed you of high kynurenolic acid, and we take only those individuals in the upper distribution of that, above 50%. And we'll perform an efficacy trial with only high kynurenolic acid biomarker subgroup using this KAT enzyme inhibitor KMD001. And we're hypothesizing that this will give us a sample where we can see an effect if an effect exists. Now with that, I will go on to talk about another study, and the basis for this study, instead of coming from BSNP entirely, comes from my laboratory. And it will really tell you a story of hippocampal hyperactivity, and what our goals would be to normalize hippocampal hyperactivity and psychosis. And these are some of the people in my lab at UT Southwestern. Here's what a hippocampus looks like. A human hippocampus is an anterior portion, the posterior portion, and as we cut it out of our human postmortem brains. We know a lot about the hippocampus because of the connection to HM, the connection to memory and memory failure in the past. There's a trisynaptic pathway within the hippocampus from the entorhinal cortex to dentate gyrus, CA3, CA1, subiculum, and then out to the rest of the brain. The hippocampus supports declarative memory in brain. There are many hippocampal alterations in psychosis, especially in relational memory. There's a consistent, even though very small, reduction in hippocampal volume in schizophrenia. There's an impairment in memory performance in schizophrenia, particularly in relational memory, that's disproportionate to the overall decrease in cognition. There's an increase in hippocampal perfusion at rest, and there's an activation deficit during declarative memory tasks. So this is just to support the idea that the hippocampus is significantly affected in schizophrenia. This shows you the human hippocampus as you would see it in an MR scan. This is the anterior part of the hippocampus. This is the posterior part of the hippocampus. It's really the anterior part of the hippocampus that's in which activity is elevated. This is the PET scan showing regional cerebral blood flow. The 0 line is baseline, is the normal baseline. There's no elevation in the middle or posterior hippocampus, but the elevation is in the anterior part of the hippocampus. We took human hippocampal tissue in an attempt to look for the molecular biomarkers that characterize schizophrenia versus healthy controls. And this shows you that shows you what the structure, again, what the trisynaptic pathway looks like from entorhinal cortex to dentate gyrus. I just showed you that a minute ago, to CA3 to CA1. It was in this pathway that we wanted to examine these different, molecular markers in these different subfields. And we were looking for hyperactivity. We were looking for what was the molecular marker of hyperactivity. The first subfield that we took a look at was the dentate gyrus. And what we found, we took a look at gluin-1. This is the obligate subunit of the NMDA receptor. And instead of finding hyperactivity, we found hypoactivity. Well, that was really shocking to us. We saw hippocampal hyperactivity on an MR scan, on a PET scan, and here we're finding reductions, but not increases in dentate gyrus. Well, we went on to take a look at CA3, and what we found was a very curious thing. And that was it. We found increases in activity in CA3, and we found increases activity in CA1, suggesting that it was the reduction in excitatory drive in the dentate gyrus which caused a reactive hyperactivity in CA3. We found increases in CA3 in the gluin-2B receptor, really substantial increases. We found increases in BDNF in both CA1 and CA3. And we found increases in PSD95, which is a marker of glutamatergic receptors, a postsynaptic marker. But then we did some Golgi. These are Golgi stains of human postmortem tissue. This is a pyramidal neuron in a person with in a normal healthy control. This is a pyramidal neuron in a person with schizophrenia. And the the mossy fiber from the dentate gyrus hits the pyramidal neuron in the supra in the area just above the cell body. And you can see that in this area in normal people, you see a few little bumps, but basically not. You basically see a fairly smooth axon. Here in schizophrenia, instead of a smooth axon, you see an axon with a lot of these little bumps. They're called TEs, and these little bumps contain an overgrowth of glutamate receptors. So then we made another model for psychosis and schizophrenia, a model that has a molecular lesion, perhaps molecular lesions of different varieties in dentate gyrus, and these are molecular lesions that decrease the excitatory drive in this pathway here to CA3, and the response in CA3 is to increase LTP, to increase cerebral blood flow, and then to pass that increase on to CA1, to subiculum, and out to psychosis. And that was our model. We took tissue then from dentate gyrus, from CA3, from CA1, and we did transcriptome analysis. And I won't show you the whole transcriptome analysis, excuse me. I won't show you the whole transcriptome analysis, but I'll show you the transcriptome analysis from CA3. We did a differential gene analysis, a WG-CNA. We looked for specific subfield-specific molecular outcomes. And in CA3, what we found was that CA3 was enriched in excitatory neuronal genes that were involved in excitation in CA3, which totally matched the model that we drew. And we found really in CA1 an interesting finding, which was a slight decrease in the enrichment of excitatory neurons, but an increase in inhibitory neurons, as if CA1 was trying to do something to manage that afferent increase. Well, this was a fairly simple lesion that we could put into a mouse, because what we found in the human was a dentate gyrus specific gluin-1 reduction. And it just happened to be that there was a dentate gyrus specific gluin-1 gene knockout in a mouse, suggesting a dentate gyrus specific NMDA receptor deficit. So we took a look at this mouse, and in fact, both in CA3 and in CA1, we found significant increases in our marker, and our marker now is CFOS activated neurons. And you can see these CFOS activated neurons here with the arrows. You can see quite an increase in them. That increase in the CFOS neurons was only in the ventral areas of CA1 and in the ventral areas of CA3, and not in the ventral areas would be the anterior areas in a human, not in the posterior areas or dorsal areas. And the other thing that we found after we found all of these significant increases in hippocampal formation, when we looked at the rest of the mouse brain, we found increases in a couple of the regions in the amygdala, in the medial prefrontal cortex, and increases in the striatum, suggesting that hippocampal hyperactivity really distributes itself around the limbic system to the nucleus accumbens, to the amygdala, and then on up to the medial prefrontal cortex. What we've noticed lately is that this whole increase, the increase of the hippocampus to dentate gyrus reduction, is really limited to the adolescent period, which is quite interesting for everything that we know about the window of risk, the risk window for psychotic illnesses, especially for schizophrenia. So we have some proposed treatments that we will try. Levetiracetam is a drug that's specifically directed to hippocampus to lower hyperactivity. Muscarinic agonists could do this. Glutamatergic drugs. We'll check out the action in this animal model and then perhaps in humans of our special antipsychotic drugs, clozapine, sulfuride. Perhaps it's neuromodulation that would really correct this. It may be a stage-specific treatment without any effect in later disease. Now, this hippocampal hyperactivity, we believe, is important in schizophrenia, is important in aspects of the pathophysiology. It may be important only in early illness. It may not be so important in later illness once psychosis is established. Now that we have biomarkers to mark certain characteristics of human brain state, we have to figure out which of these biomarkers is really important in schizophrenia. That's exactly where I wanted to end. I wanted to say that these are all hypotheses. These translational biomarker fingerprints remain to be established and validated. These are hypotheses. These are not these are not known. We need data to show which biomarkers are informative to predict treatment response, like the first study I showed you, to predict outcome prediction, or that marks molecular disease targets. Different studies may require different approaches, as you saw me outline today. Will this be useful? You're going to say, well, will any of these studies be useful? Will targeting biomarkers for treatments be useful? The answer is stay tuned. We don't know for sure, but we've done as much as we can with a phenomenology, and we think that moving to biomarkers will give us added information. I have had many collaborators in all of these studies, collaborators in my own laboratory, BSNP PIs. Dr. Schwartz's name is not on here, but he's been a collaborator in these studies as well. And I thank you very much for your attention today.